Good afternoon or good evening, wherever you happen to be in the country. I'm in Central Coast, California, and so it's 4 o'clock for me. My name is John Molinari. I'm a microbiologist, and I want to thank Kerr for sponsoring this webinar, this update on COVID-19 vaccination. We are going to be talking for about 45 minutes, and then I want to leave it time for questions at the end. This is going to be a brief session, but I really appreciate your taking time out of your days to hear this update, and hopefully I'll be able to provide you with some useful information. As we go through tonight, these are the kinds of things that I'm hoping to accomplish with you. Want to show you about the historical importance of vaccinations in population health. We're going to describe current science-based status of COVID-19 pandemic. That's going to be very, very brief, just a couple of slides. And we're going to spend most of the time on the COVID-19 vaccines, looking at recommendations, effectiveness, success, and very importantly, looking at the last aspect of what we're going to cover is how the emergence of the viral variants can affect vaccination success. This is not going to be necessarily in this order, but we will be covering this information this evening. I typically start with this slide when I talk about vaccines with my students or groups at meetings. And this slide is something that I put together from CDC data that have obviously been developed over decades, showing the impact of vaccines in the 20th and 21st centuries. One of the things that is important to note is that at the end of the 20th century, the CDC published a document, if you will, that described vaccination, public health vaccinations, as one of the 10 greatest public health achievements of the 20th century. And you can see some aspects of that achievement in this slide. One of the things that I typically do when I'm talking about the slide, and I wish I was with you in the room so I could have you do it, is I would ask participants to look to the right, look to the left, look around them, and I say, if it wasn't for the vaccination that your parents and grandparents chose to receive, some of you wouldn't be here today, because these diseases not only have a morbidity, but they have a certain percentage of mortality, and it used to be far greater. We don't even talk about chickenpox, for example, anymore. You see down the bottom, varicella. Now we talk about shingles. Smallpox is the only infectious disease in the world that we have completely eliminated. That was 1978, I believe. We are very, very close to eliminating polio in the world. We haven't had a case of polio in the United States, a wild type case of polio in decades. We've had a couple of outbreaks of the infectious diseases in recent years other than COVID. Measles comes to mind in 2019. But look, with the success of vaccinations, as of October of 2021, early October 2021, we had 37 cases this year. Look at what we used to have for the average in the 20th century. We have changed the face of infectious diseases with public health vaccines. I had friends. I had friends in grade school, in grammar school, that had survived polio. This was in the 50s. They had survived. They had damage to their legs, to their limbs, but they were alive. We had a family around the block who lost a child in an iron lung. The younger people listening to this don't even know what an iron lung is, but the older people do. Look at how we have overcome so many of these types of widespread infectious diseases that used to ravage the population. Many people don't realize anymore, especially the younger people, with German measles. If a pregnant woman contracted German measles, there was a real risk of her baby being born with congenital rubella syndrome. And that could lead anything from damage to the brain, damage to other organs, the baby being born dead, or thank God in most instances, the babies were born okay, but there still was that risk. And as I said, we don't even talk about chickenpox anymore, but the year before we had the chickenpox vaccine in 1995, we had over 4 million cases, and people were hospitalized and people were dying from that, typically young children. we have been able to accomplish mass vaccination in the United States. These are three major ones with diphtheria, polio, and measles. Some of you who are more mature in your age, like I am, you can remember the polio vaccine campaigns, certainly the measles vaccine campaigns in the early 60s, where we really cut down dramatically on the incidence of the infections in the population. It was a personal responsibility, obviously for our personal health, for our family, and also to help protect other people. And we did it. And look at the success that we have been able to achieve with these. When we look at vaccine development, there are some absolutes in this. And I say absolutes, and I have to be very careful, and I apologize for using that word maybe, because no vaccine is 100% foolproof, as you see on this slide. But the absolutes are, you have certain ideals that every vaccine has to strive to meet as much as possible before they're ever even really considered for final approval to be released into the general population. And first and foremost of all of these is safety. Vaccines have to be far, far more safe to the people that are going to be receiving them than the detrimental effects of the disease itself. So when you're looking at side effects, the safety of these has to be very, very much overwhelming, where the benefits of the vaccines far, far, far outweigh the potential adverse effects, typically most of which are very mild and common. Obviously, a vaccine has to be antigenic or immunogenic, has to stimulate a protective host response. That can take years of work to find the protective antigen or groups of antigens. And you see the other features that we look for in these. A big one, especially in today's world, is that it can be administered to immune competent and immune compromised individuals. In our population, we have an increasingly greater percentage of our population is immune compromised. People who are living longer with conditions that they used to die from. Cancer survivors, transplant survivors, diabetics, lupus, rheumatoid arthritis, people on prolonged steroid therapies, the elderly. This gets very depressing, by the way. By definition, 65 and older is considered elderly, and according to public health calculations and all, elderly individuals are much more susceptible to a variety of conditions which can compromise their immune defenses. So we look at these safety factors as being absolutely essential with this. We have had, and I'm going to get into that in a little bit, we've had a number of vaccine platforms. I'm not going to bore you with all of this, but if you look at inactivated, attenuated product component vaccines that we have utilized in the population, most of you have received samples of each of these probably. The fastest growing group of vaccines would be the component vaccines, where actually scientists spend time, a lot of time in some instances, finding the specific component of an organism, a virus, a bacteria, where we can develop a protective immune response against. And then it's the charge of that and the challenge to isolate that specific antigen and utilize that in vaccines. If you have to look at the COVID vaccines, you're looking at the spike protein as the major antigen that has been utilized as the target, if you will, for an immune response in our vaccines, in our vaccine responses. And so that type of information is very, very useful, because you want to make sure that you have an immune response that's as effective as possible for the specific diseases. We have come a long way over the years in utilizing science and genetic science to develop other approaches to maximize vaccine safety and efficacy and effectiveness. For example, a number of you listening to this, you actually have received a recombinant DNA vaccine for hepatitis. You received the Engerex or Recombavax. people like me, we received the original Heptavax B in the early 1980s, but in 1986, it was replaced with these recombinant viral DNA vaccines that they used altered yeast cells to produce the hepatitis B surface antigen. So you've already had genetically modified vaccines introduced into your system, and you responded well. The Gardasil with the L protein particles into spheres, work with recombinant nucleic acid studies for that led to the Ebola virus vaccine. Not that you've received Ebola virus vaccines, but these vaccines have been very effective. And now, as we've had another outbreak of Ebola in a couple of countries in Africa, in West Africa, these vaccines are being very, very well utilized to control that epidemic, unlike what we had before we had the vaccines a couple of years ago. So these nucleic acid vaccines that you see here, DNA messenger RNA, that were experimental before 2020, really have had a whole lot of history with the advances in science. I did want to spend a couple of minutes with you before I get into the vaccine development to update you just a little bit as to where we are with the pandemic, a little bit on vaccines, but also want to talk about the variants. And then we'll get into later on as to how they can affect the effectiveness of the vaccines. As of a couple of days ago, we still, thank God, are seeing a decline in cases. It's very uneven depending on what part of the country you're located in. I'll show you that in a little bit as well. But look at the bottom. Over 712,000 people have died in this pandemic, and that's reported deaths. There may be more, but the figures are probably closer here in the United States than other countries. Other countries have been severely underreported for cases and deaths. But this has been, obviously, as you've all heard many times, the worst outbreak, the pandemic, since the Spanish flu in 1918, 1919. What is alarming to me and to many of us is when you look at the vaccination incidents in the United States, and I'll show you how we are lagging in so many other industrialized countries in the world. One of the effects on vaccination, of course, has been due to the SARS-CoV-2 variants. Just as an introductory statement, SARS-CoV-2, of course, is an RNA virus. It's a respiratory virus, and RNA viruses tend to mutate at a faster rate than DNA viruses like hepatitis B virus. Also, as a respiratory virus, it can be much more readily transmitted from host to host, including animal hosts and human hosts. And one of the things about these viruses is that the more hosts that they have to find, and they can find, the better the chances they have of having these mutations occur, most of which don't affect infectivity or severity of disease, but which can. And with this pandemic, we have seen in this country 4 SARS-CoV-2 variants of concern, which is where we have had significant numbers of cases. And so we have to do much more genetic mapping and, of course, try to control them. The World Health Organization has added, I think, U to that list. We haven't added mu to that list. That's the most recently detected variant. But you see the ones that we have had to deal with. And you'll notice down in the middle of this slide here, increased transmissibility from the Alpha, which is the one that was first reported in Great Britain. And then we have the Delta, which was first reported in India. But typically they give them these specific scientific classification names. Notice immune evasiveness, which talks about how the binding sites for the antibodies may have been altered on the virus and make them a little bit more resistant to host immune responses. It's important to note that with all of the variants that we have as variants of concern in the United States here, vaccines are still overwhelmingly effective. Very high effectiveness compared to obviously, some other vaccines which are not nearly as effective. Influenza, for example, is one that has never even come close to what we have for this virus. Where we are with the variants has also been changing. And I want to show you this slide, actually two slides. This is as of the end of May, where we had the tracking in the United States of the variants. And as you can see, what started in the middle of March here, we had a number of these strains or variants, I still call them strains, of SARS-CoV-2. But the B.1.1.7, the one that had originally started in Great Britain, that was more aggressive and seemed to not react as cause a little bit more severe disease, more infectious, was gradually taking over in the population until by the middle of May, it had pretty much taken over the majority of the cases. It was about 69, 70% of the cases. I want to point your attention to this little small area down here, this little magenta area right here, because this is one that we see, which is the B.617, and that's the Delta right here. That's the Delta. It was a very small percentage of cases. But look, as weeks and months went on, B.1.1.7 declined, and look at the B.617 increasing. This is the Delta, and look, the Delta now accounts for over 99%, over 99.8% of the cases that they have figured out the genetics of the virus. So it is overwhelmed. It is far more infectious and causes much more severe disease, especially in those who are unvaccinated. I'm going to get into that in a little bit. Where do these variants come from? What really are they? As I said, it really goes back to that spike protein that I talked about. And here is an early set of diagrams that were published early on in the pandemic, looking at three-dimensional models or the stick proteins, diagrams for the receptor binding domain for the spike protein. Up here, this is where it's down, not active. Here's where it's up. This is where it can bind to the ACE2 receptor on host cells. Virtually all of the host tissues we now know have receptors for this virus. So it used to only be respiratory, but now we know it can spread throughout the body. And here as comparison, you see the RBD for the original SARS-CoV-1 back in 2002, 2003. And you'll notice the difference. This was a more lethal virus. Thank God we controlled that to less than 10,000 cases, but this is a far more infectious virus. And when you look at how the mutants have changed, the mutations of the virus have changed, what they have done is they have modified and deleted and various amino acids in the proteins, and they have included other amino acids which have changed the configuration of the receptor binding domain of the spike protein. This is Delta, and you'll notice the number of modification sites on Delta compared to the earlier variants that we saw. This is one that has caused Delta to be much more infectious. World Health Organization back in June made a statement, said it's faster, fitter, and will pick off the more vulnerable more efficiently than previous variants. Antibody effectiveness as far as cross-reacting antibody is not 100%, but still the vaccines are very, very effective against this. But when we look at its spread, and you saw in the previous slide just how far it's spread, is over 225 times faster in its infectiousness than the original virus that started in 2020. So that makes it much more efficient at attaching and invading cells. It can spread throughout the population. I'm going to show you that in a little bit. It's actually considered to be as infectious as chickenpox. Chickenpox is an aerosol-mediated disease, can be transmitted by droplets, but primarily aerosol-mediated. And this virus, this mutant, this variant, produces many times more viruses than the other variants. which now makes it able to overwhelm some of the reactions, responses that we've seen to vaccines, but certainly overwhelm hosts and be far more dangerous to people who are unvaccinated. I think 98% of the hospitalizations and over 99% of the deaths from COVID-19 in recent months have come from unvaccinated individuals, which of course then talks about the importance of vaccination. This is the diagram I talked about as far as its infectivity. Here is the original ancestral strain, and you see it's infectious. It's not the same as swine flu, 1918, 1919. Not as infectious as SARS or MERS, the earlier coronavirus outbreaks that we saw in the early part of the century. Then we have the Delta variant, which is far more infectious, and it is analogous to chickenpox. Measles is far more infectious than SARS-CoV-2. It has an RO of about 16 to 18, an infectivity factor. The Delta variant is about 5 to 8 compared to 2 or 5 for the earlier variants that we saw. So you're seeing that. Bottom line is, as we have people who are not protected, this virus has a number of hosts that it can still infect. And the more it can infect different hosts, the greater the opportunities it has to mutate and to come up with different types of variants or strains, as I said before. If you want to think of a simpler non-infectious example, think of influenza. We need a new flu shot each year because the strains continue to differ each year. They have these point mutations, and this is what you see with SARS-CoV-2, except this is far more dangerous, has a much higher incidence of hospitalizations and deaths, and the variants have become much more severe. Each of the surges that we've had with this pandemic has been due to an increasingly virulent and increasingly transmissible and increasingly serious variant of SARS-CoV-2. It makes sense then that the work on the vaccines would focus on spike proteins. Understand that this work was not something that was initiated in 2020. This work was initiated as a result of the original SARS pandemic in 2002, 2003. I know we hear people talk about, well, this is so new, we don't know a whole lot about this, what's the data for this, et cetera, et cetera. Studies were done to develop a SARS vaccine, a coronavirus vaccine, back in 2003 to protect against others. And so the basic genetic work, the basic virology work was already in place. And originally, you see the FDA for something like influenza viruses and their vaccines, they were looking at 50% effectiveness for possible approval. Well, obviously, they were looking at much higher for this because we had a very specific antigen. And you see here's the receptor binding domain of that spike protein. The SARS-CoV-2 vaccine was developed, thank God, in 11 months, in less than a year. And that's important. But please, to reiterate, understand that it's not that we started working on a vaccine with starting from scratch. As I said, this work had started back in 2003, and there were multiple laboratories, international studies, reports, vaccine development for the original SARS outbreak. So the work for the coronavirus vaccines is not new. It's just that, thank God, you had a mobilized effort by literally every country in the world, and thousands or hundreds of companies put all their resources together to come up with different types of vaccines. Messenger RNA vaccines are one of the ones that really work, and that's the ones that we have in our country with Pfizer, Moderna, and J&J. This is an important slide because you hear people talk about the safety of this vaccine, and remember what I said about safety being absolutely paramount. Well, this is what we talk about for safety. There are 6 basic phases in vaccine development before the FDA and Advisory Committee Immunization Practices gets the information from the pharmaceutical companies to evaluate. The first three phases are preclinical trials. And this is where you have all the laboratory studies. You have to have the scientific studies to see if there is a very specific antigen that is going to stimulate an immune response from the virus, much like hepatitis B and much like hepatitis B surface antigen for hepatitis B vaccine. And so we were vaccinated with hepatitis B surface antigen. Well, that takes a while, and that's taken a number of years of effort to figure that out. You have laboratory studies in animals to see if they would respond to an inactivated virus or an attenuated virus or the protein, what dosages, what were the adverse effects in the virus vaccines? Did the animals develop convulsions? Did they develop allergic reactions? Did they become lethargic? What are all sorts of things? What is the best dose per body weight? Looking at these in a variety of animal models. So all of this is preclinical. Then you have the three clinical phases, but they each depend on the earlier one. The phase 1 clinical trial is where you have a relatively small number of people. You have had the laboratory data, the preclinical trials approved. So they have an IND to do the investigation in humans, and you have volunteers for this. And these are studies over a couple of months to look, to see if the people develop COVID that have been vaccinated versus the placebo group. Did the vaccine group have any sort of adverse reactions? Were they mild? Did they have anything severe? Did you see allergic reactions? All those types of things to look at not only the effectiveness of the vaccine, but look to see the responses of the host to the vaccine in other ways. Then you have phase 2 and phase 3 in increasingly larger populations. Phase 3 would be tens of thousands of recipients of the vaccine. And of course, you have a similar placebo group. And there, with each of the increasingly large groups, you're not only looking at vaccine effectiveness, but you're also looking, because you are vaccinating more people, to see if you have the possibility of some rarer adverse event that you hadn't picked up in the smaller groups. The larger the number of the groups, the better the chance you have of picking up rare adverse effects. And they had picked up some. There were some allergic reactions, as I recall, but these were then considered not to be severe compared to the effectiveness of the vaccine. All of these have to be looked at, as I said, with safety in mind. Safety in mind. The Advisory Committee Immunization Practices gets this, reviews it, and the FDA makes a final determination based on the accumulated data, which has taken quite a while. Even after a vaccine is released to the population or to different age groups and gradually to the population, it doesn't stop being monitored. Notice safety continues even after the vaccines have been released. And this is where you have these vaccine adverse reporting systems, the vaccine safety data leak. You have these systems in place to continuously monitor in the population the reactions and responses of the population. Many of you, when you received your vaccines, you got your VAERS thing on your phones, so you could report if you developed a low-grade fever, if you were fatigued, if you were tired for a day, or if you had an allergic reaction. Remember, there are some individuals early on who developed anaphylactic reactions. Thank God, not life-threatening, but that was reported, and there were very, very few cases, but they were there. But they look at continuing to monitor the safety of the vaccine as well as the effectiveness. And it is possible, and it has happened, where a vaccine can be recalled even after it's been studied and tested and it's been released to the population. And then they find, because of millions of people receiving it, that they have had a problem. This happened, for example, in the late 90s with the rotavirus vaccine for infants. Rotavirus is a common infection, was a common infection in young children. And when they developed the rotavirus vaccine, what they found within less than 2 years was that more children were developing stomach problems, GI problems, than they had expected in the study groups. And so it was withdrawn. So this is not to say that when vaccines are approved, they're released, and that's the end of it. Safety continues to be a major factor. When they are tested, we look for efficacy. These are in the study groups. These are clinical trials where participants are selected. They may not have underlying medical conditions. They may not be immune compromised. They may be of different age groups to look to see if the different age groups respond. But typically, they're not going to have small children. Those are reserved for very special clinical trials. As you know, those are going on right now. And thank God, it looks like the vaccine manufacturers are close to having information to get young children vaccinated against COVID-19 as well. But these are the clinical trials. And this is the degree to which a vaccine prevents disease under ideal and controlled circumstances. Then you look at effectiveness, and this is how a vaccine responds in the real world. And now we look to see, when you're giving it out in the general population, you have people of different ages, you have people who may or may not be immune compromised, you have people on different medications, you have to look at vaccine storage. There have been rare, thank God, rare instances where some batches of vaccines had to be discarded because they weren't stored at the right temperature for long enough, or the temperature rate was raised in the facility, and they had to remove them. This has led to a series of studies, reports that have come out for effectiveness. These continue to come out as centers with multiple sites. For example, different countries have now hundreds of thousands of individuals, even millions of people, where they can coalesce the data to look to see effectiveness as well as side effects. Remember, sometimes for different infections, you may not have to have 90, 95%. For influenza vaccines, 40 to 60% was about the highest that we could get for the longest time. And yet that effectiveness saves thousands of lives every year, tens of thousands of hospitalizations, something that we really need right now as we're entering winter, fall and winter, where we're getting into flu season, another respiratory virus, not as infectious as SARS-CoV-2, but yet another potentially complicating factor as to how we have to get COVID under control, because now we're entering another respiratory virus season. There have been studies, as I said. This was one study that came out. I want to say this was Mayo Clinic, but I could be wrong. But notice they had a 35-week study period, and they found that overall the vaccines, COVID vaccines, were 80% effective in preventing SARS-CoV-2 infection. Remember, SARS-CoV-2 vaccines prevent infection, and that's great. But they also, most importantly, protect against hospitalizations and death. And we'll talk about breakthrough infections a little bit later on. Even with the Delta outbreak that we still are going through, the effectiveness in some sites went down to about 2/3 of what it was originally, but still, that's very, very high overall, 80%. And remember, these things started out 90, 94% with the original strains, but each variant has had its own characteristics, which can complicate something. But the basics are the vaccinations work. What do we look at as far as side effects? I hear people talk about, well, I'm afraid of side effects in this. Well, we've all gotten vaccines, and the overwhelming majority of adverse effects to COVID-19 vaccines has been mild and transient. These are some little common ones. We've all had family members and friends. Maybe you yourselves have had these, and they're transient. You don't feel good for a day or so. I fortunately, I didn't have any real adverse reaction. I think I had like a little itch at the injection site for a couple of hours, but nothing. It was, and that was about 12 hours later. But again, these are the kinds of things, and even more severe reactions if you had them, that you can report so that they can be evaluated and looked at to see if there's something going on that they only see when millions of people get the vaccines. There was a large study that was published in April, looking at over 3.6 million individuals that have been vaccinated with either Pfizer or Moderna. And they looked at injection site reactions and any site reactions. This is injection site reactions. I know it's a little small, but you can see where you fit in. This is for both vaccines, dose number 1, both vaccines, dose number 2, and then they break it down to the individual vaccines. And you see any site injection, 65% for Pfizer, 73% for Moderna. Notice how the figures went up for the second dose. Well, understand, by the time you got the second dose, you already hopefully had started to develop an immune response. And so you have inflammation into the site of where the foreign antigen was injected into the system. These types of reactions are transient, they're not unusual. Remember, you are injecting a foreign antigen into your body. And the first part of your immune response is acute inflammation to the site to phagocytize and begin the process of immunologically producing antibodies and sensitized T cells later on. So see where you all fit into these, okay? Then you look at any systemic reaction, and a lot of people had fatigue and headaches. That was probably the most common one following the first and second dose. Notice fatigue after the second dose with Pfizer and Moderna was much higher than fatigue after the first dose. I heard this many times from family members and friends. And you see a fever was less, but it was higher for the second dose. Okay, 7% first dose, 21%, then 10%, 36.7%. And you can see where, if and where you fit into any of these. Also, studies had to be looked at when the vaccines were released. And again, you have millions of people receiving these, as the rare untoward effects that maybe they didn't pick up or weren't able to pick up in some of the clinical trials to the extent that they did later on, like myocarditis. And they had cases reported of this. But when they looked at the cases, these were about what was expected in the general population. They weren't cases that were abnormally high, that could have led to severe health problems or possible hospitalizations or things like this. All these things have to be reported, and all these things have to be analyzed, and they were. And so the vaccine continued to be far more effective than any potential adverse effects that they've looked at. So of course, this has stayed on. I put this on here. This is as of a couple of days ago, because this is to me, it's not good. We as a country are 56%, maybe 56.2% fully vaccinated. I got this slide and I put the numbers in. This took a while, but I found out I missed a slide. I missed a state. I missed Hawaii. I'm sorry, Hawaii is like about 60, let me take a look, 59% fully vaccinated. I apologize to anybody who's listening from Hawaii. I can't believe I did that. But you can see where your state is for fully vaccinated. And we have some states still in the low 40s, even though you may have 59, 60 some odd percent in certain states. We still have sections of the country where the vaccine rate is not as good as it should be, different counties, and you can get that information as well. I want to show you something which shows you where we stand compared to the rest of the world. And this is a couple of weeks ago. This was toward the end of September. And I put this slide on because I have a more recent slide, because I couldn't find Canada data recently in some of the other countries that are on here, but I want you to see where we are, where we were, compared to other industrialized countries and a non-industrialized country like Brazil that still is lagging. But you look at people that have at least gotten one dose. A couple of weeks ago, we were about 64%. Look at the world, India, and then you look just a couple of days ago, and again, you see the disparity between countries with fully vaccinated, at least one dose. And then you look at the United States. We still have a ways to go. Originally, we were talking about 70% immune vaccinated for herd immunity. That figure has been delayed and increased dramatically because we have been slow for this, and we are lagging behind, and it's sad. If we look at information that's come out from our world in data, also Kaiser Foundation has done studies with people who are willing to get vaccinated, people who are not willing to get vaccinated. You can see various countries where they stand for vaccinated, willing to get vaccinated. You see here, this is about 63, 60, about 65% now with one dose, the most recent data. But you see here, people willing to get vaccinated. And you see that we still, if you look in the red area, we still have a significant percentage of our population that is still unwilling. And this is where you're getting the mandates. I really wish we didn't have to do that. Growing up, we never had anything like this. It was our responsibility to protect ourselves and to protect each other. And somehow this has been changing. I apologize for saying stuff like that, but it bothers me a lot. By my estimation, looking at the population of the United States, we have over 112 million individuals in the population who are at this point not vaccinated at all. That's a significant percentage. That's over a third of the population of the United States. So we have a ways to go for this. If we look at reasons why vaccines have not been received everywhere, you can see this list. A big one has been concerns about side effects, long-term effects. I've had questions on these. I'll get into that in a little bit. People saying it was so new. I'll talk about recent COVID-19 infection, the reason why we don't want to get to herd immunity this way. We look at perception of lower risk in certain individuals, younger age groups, late teens to 30s, early 30s, have been lagging in vaccination as to the fact that they early on didn't think that they were as affected by this. It was more of a mature person, older person, and that's too bad. There have been a lot of talk about pregnancy and wanting to get pregnant. The CDC has come out with recommendations showing that, in fact, pregnant women are at much higher risk for having problems with COVID-19, and the babies are at higher risk. And there is not a contraindication for pregnancy or a woman who wants to be pregnant to get vaccinated. And then we get into something like this, and this gets, again, people think of things. Understand that there are vaccines that we have used for decades and decades and decades, and there can even be with vaccination rare, severe reactions. Yet we have done these. Many of you received the oral polio vaccine, the original Sabin vaccine, and it was an attenuated virus. And in some instances, because we didn't know if a child or an adult was getting the vaccine was immune compromised, there were rare instances of them developing vaccine-associated polio paralytic myelitis because the virus was able to go from the GI mucosa across the blood-brain barrier and cause polio. That's why they replaced it with the Salk vaccine, again, the original vaccine back in 2000. But millions and millions, including many of you, received that vaccine. There was a rare risk of Guillain-Barré from influenza vaccination. But the risk is much, much more likely if you get influenza. And we've accepted these risks all along. We look to protect the general population. And this is where we get to herd immunity. So we need to reach that threshold. And originally, as I said, it was 70%. Where we look at vaccinations as a way to achieve this, if you have a population like we had before the vaccines, you had people who are healthy but not immunized, and you have people who are infected with the disease, SARS-CoV-2, COVID-19. And you see how infectious this virus could spread to where now you have many more people in the population infected and ill. When you have some of the people vaccinated below herd immunity, you have more people that are protected, but you still don't have enough to protect those who are not immune from the person who is infectious. And with Delta, it's even more infectious than the other variants. The result is you still have spread of the virus to the population. The key is to reach a level of immunity in the population where now you can protect those who aren't vaccinated, those who aren't immune, maybe those who can't get the vaccine because of their health condition or some other factor. But now you have a significant percentage of the population that's immune, where now they can shield these who are not able to become immune on their own with the vaccination, and thus you have control of the virus. We have looked at this. There are two ways to achieve herd immunity. Vaccination is by far the most advantageous artificial active immunity. Some of you have had COVID and you've recovered, so you've developed natural active immunity. And the call has been, well, why can't we just use natural active immunity? Well, natural active immunity works in a number of infections and can provide lifelong immunity. But this respiratory virus and these groups of respiratory viruses that have this capability of mutating so readily as they go to different hosts can change very quickly, as we've already seen with the number of variants that have come out. We don't know how long protection lasts. With influenza, it doesn't last a couple of years because there are different strains. And so there's not cross-reacting antibodies. And so we have these variants that have different modifications in their antigens, and the antibodies may not protect against some of these variants. So we need to continuously look at vaccination, which is the broadest sense of development to induce immunity. What some people have forgotten about also is the impact on population health and healthcare systems. You're all aware of long COVID. We started seeing this early on in the pandemic with respiratory damage, people having problems with taste and smell, mental fog, that's called brain fog, and there's all sorts of tissue manifestations. Well, it's been estimated that 1 in 3 COVID-19 cases develop these long-term health issues that could go on for weeks to a couple of months, and some reports have been even 9-10 months. But at least 1 in 3 have some sort of prolonged health issues. And these could be, as I said, fatigue, cough, brain fog, damage to multiple tissues and organs. We've seen damage to kidneys, livers, hearts. It's a problem. And it's been seen in all age groups. So if we were to rely on natural active immunity, and we still have over 112 million individuals not even receiving one vaccine, what does this say as far as long-term pathologies, deaths, and our hospitals that have been struggling mightily, as you can see when you read look at the news at night? I told you also no vaccine is 100%. Why? Not everybody responds to vaccines. About 10% or so of people don't respond to hepatitis B vaccine. I didn't respond to hepatitis B vaccine with my first series. I had to have a second series. My students thought it was hysterical. I was one of the first ones to get it in the summer of '82. So we don't know. We don't have a good test to see if the immune response that you have is going to be protective. Is the recipient immune compromised, where they're not going to respond as well? Did a recipient
develop a weak immune response, especially if they're immune compromised or elderly, where they don't have a senior dose, as we have with something like the Pneumovax. Was the person asymptomatically infected at the time of vaccination? I know they check the temperature and ask if you had symptoms, but maybe you weren't. Remember, at one time they were talking about over 30% of the cases being transmitted from asymptomatic individuals. And then you look at the variants again, and depending on the concentrations of viruses that are produced by these variants, depending on the cross-reaction of host immune responses from vaccinations or even previous COVID-19 cases, with the current domain dominant variant, which is Delta, that can have an effect. That can have an effect. So we look at what is the effect of COVID, or what is the effect of vaccinations on these? And you look at the vast majority of COVID-19 hospitalizations and deaths occur in unvaccinated. I told you the percentage earlier. And the vaccines are designed to prevent severe illness, not infection, but they do definitely protect against infection in the overwhelming majority of cases. But there can be breakthrough cases. And we have seen breakthrough cases, and we've seen breakthrough cases in people that received vaccines. And you have to understand this happens. A number of you received influenza vaccines in previous years, and you got the flu. You may not have responded. You may not have responded to a specific strain that was in the population. So this happens, but it's far more severe for the infection to occur in unvaccinated or someone who's severely immune compromised. Question I will leave you with is, do we need boosters? My opinion, yes. There was never a doubt that we're going to need boosters because, again, these RNA viruses are known to mutate and cause variants. We've seen these. SARS-CoV-2 does have a high rate of mutation. I saw literature a couple of months ago from global studies and World Health Organization getting data that SARS-CoV-2 since the beginning of the pandemic, I'll say January of 2020, has mutated over 12 to 13,000 times. Again, most of those mutations are not anything that's going to increase its virulence or transmissibility or affect the course of the infection, but you've seen where it can. And each of the waves is caused by variants which possess immune escape properties and have been more severe. And SARS-CoV-2 is not a seasonal flu. So far it has gone through all 4 seasons, and it's not going to go away. And SARS-CoV-2 is not going to be the last respiratory virus that we're going to have to deal with. So this is something that we have to look at because it does affect virtually every tissue and organ in the body. And we need to slow down viral spread, replication, and mutation. There have been recommendations for boosters, and you see these listed here. And they're looking at adding more groups to the vaccine groups, as I said, young children, and also with the boosters as well, to augment the primary immune response that we've had already to protect us. At this point, I want to end it and open up the questions, but I did want to thank Kerr very much for providing sole sponsorship for this seminar. Really appreciate the effort that they put into helping put this together. And at this point, Delaney, I'll be happy to answer any questions that they have.
All right, yeah, it looks like we have some questions. So the first one, have they ever mass mandated a vaccine like they are with COVID?
I'm sorry?
Have they ever mass mandated a vaccine like they are with COVID?
No, because people responded. They didn't have to. Somehow this has become politicized, and I'm not going to get into politics because that's not the point here. But the people responded. I can remember as a kid, the nurses coming in and vaccinating in schools. I remember there were whole groups of individuals getting together in small towns and having nurses and physicians coming in and vaccinating at a school or something like this. it was just a personal responsibility for our health, our family's health, and for the public health, for other people.
All right. The second question is there a traditional attenuated vaccine for COVID planned or only mRNA?
There have been studies on attenuated viral vaccines in other countries. I didn't put that slide on, and they haven't been as effective. They haven't been as effective to my knowledge. But there were studies done on attenuated vaccines. There were studies done on inactivated vaccines as well. They haven't been as successful as the messenger RNA vaccines.
Okay. And the next one, is there a correlation between immune response to a vaccine versus the reaction that happens right after? In other words, if I reacted strongly with many symptoms to the SARS-CoV-2 vaccine and my subsequent immunity.
Okay. You mean like if somebody has, I think the question is saying, if someone gets the injection and they have severe injections like soreness, they're fatigued, they have a low-grade fever, are they going to respond better? I believe that's the question.
Yeah.
And no, that's not necessarily true. There are people who have a much more acute inflammatory response, or at least at that site, it seems to be greater. Remember, the first response is going to be acute inflammation. Some people may not have as much tissue at the site of injection, and so the inflammation can seem more severe, and they may develop a fever much more readily than others. I've seen people who had very, very little tissue responses, if you will, and they have very high antibody titers. This is like the hepatitis B or Pneumovax or some of the other vaccines. And so I don't believe this is necessarily a correlation. And some of it is perception. You know, please forgive me, all of you. Some people think that, oh my God, my arm is falling off, and you know what? It's not that bad, but there isn't any correlation that I'm aware of.
Okay. The next one is, how do the vaccines which are offered here in the USA, as in Pfizer and Moderna, rank in terms of the other vaccines in other countries? When international travel opens up more, could boosters be a possibility to enter other foreign countries?
Okay. I think what you're saying is, how good are the Pfizer, Moderna vaccines compared to other vaccines in other countries?
Yes. Yep.
I think they're very good. I can't give you a numerical rating, but they are very good. You had the J&J vaccine, the AstraZeneca vaccine in Europe, which was released first in Europe. They also work. I haven't seen an international ranking, but the Pfizer and Moderna, when you look 94, 95% as a result of the clinical trials and the high effectiveness that they still have with Delta, they rank very high. I haven't seen that information though, as far as ranking with the other vaccines. I apologize.
All right, the next question. Why does the vaccine protect more than the natural immunization after a real infection?
Good question. Well, when you are infected with the virus, like this one, you develop antibodies, okay? But it's going to be your own immune system that's going to develop the level of antibodies to that infection. As I said, some people develop low titers of antibodies, and I'm just talking humoral immunity. I'm not talking cellular immunity. So some people may develop high titers of antibodies. Other people may develop low titers of antibodies. And so you're not really quite sure. With the vaccines, they have at least data from the clinical trials and information that they've done with post-clinical trials to look to see the types of responses that can occur. But a natural infection, especially when you have an individual who may not be a good immune responder, can lead a very transient response. I don't know, that's maybe a few months. Other people may last 8, 9, 10 months. So it's difficult to catalog that way. You would have to have really long-term good studies to look at the titers of antibodies. I can tell you, for example, with hepatitis B, we used to do screening tests on our students and faculty and staff who got the hepatitis B vaccine to measure antibodies. And there would be antibody titers from A to Z. Some people would have low antibody titers, other people would have high antibody titers. I told you I didn't respond after the first series, so I had to go through the whole series again. And I developed high antibody titers. So you don't really have a correlation with each person, with each person's individual immune response. It's your individual immune response. I probably went too long on that.
We could, it's 8 p.m. right now. We could do, we have more questions if you want to keep going.
I'm going to leave it up to Tron. He's in charge. But I'll ask a couple. I'll answer a couple. Sure.
You guys can go
Okay.
for another 5 minutes.
Okay. What have the studies shown in regards to the COVID virus being transmitted through dental procedures that create aerosols?
I suspect most of the people listening to this are dental professionals. I'm proud of you guys. Dentistry, as you know, was listed as high to very high risk for COVID early on in March of 2020. Hygienists, dentists, assistants. And I have not seen, since you all responded with the respirators, the air recirculation units, all the things that you're doing with the HVAC systems, all the new equipment that you've had to invest in, and procedures that you've had to do now. I have not seen instances of higher COVID cases in dental settings. Maybe I'm just not aware of it, but I'm proud of you. I think dentistry responded, and I want you to keep responding, because just because you got vaccinated, this is not something to say, okay, well, I don't have to do this anymore. But I think that dentistry responded, and I think dentistry responded well. I spoke to tons of dentists and hygienists and assistants, not just in webinars, but people that I've known over the years. And the overwhelming response, I thought, was doing the right thing, as I believe that you have always tried to do.
So it looks like I think we can end it here.
Okay.
The,
Thank you all for being here this evening. I really appreciate you taking time out of your lives for this. Hope it was useful.